Hello everyone, in this video I'm going to be talking about something called lipids. Now lipids, or fats as we know them to be, I've sort of done at this level to a degree in Key Stage 4, but this video is aimed really at Key Stage 5. So we're looking at A-level biology when we talk about cells and cellular structures. Now there's a reason why I've done the I of the title in the sort of funny way that I have. It's because that relates to something that we'll talk about in the video, the structure of what's called the phospholipid. Plus I'll jazz it up a little bit. So let's think about lipids in general and the role of lipids or fat. Now when I do this bit with my students, I tell them to think of EWIP. E, W, I, and P. And this is what helps me remember the role of fats. So E stands for energy, because lipids act as energy sources when they are oxidized. Act as energy sources when oxidized. So that's what the E stands for. Now, gram for gram, you can actually get more energy from lipid than you can from carbohydrate. The W stands for waterproofing. So it acts as a waterproofing layer, if you like. And that's because it's insoluble in water. So because it's insoluble in water, because it doesn't dissolve in water, it acts as a waterproof layer. And a good example that we can think of is the waxy cuticle in plants. The upper waxy layer that we have in plants is made of lipid. The I stands for insulator, because lipids can act as insulators, because they are slow conductors of heat. That's really important to mention. They're slow conductors of heat. So the E is for energy, the W is for waterproofing, the I is for insulators, and then finally left with P. And the reason why lipid or fat is really important, some people argue or just think that fat is terrible and that we should have cut out completely all fat in our diet, and that's incorrect. You need a certain level of fat because it will surround many of our vital organs, protecting them. So the final letter P is for protection. That lipid provides a layer of protection around our vital organs. If you look at the heart, for example, if you were to dissect out a heart, you would see a very thin coat of fat around it. So that's our final letter, our P, protection. So E is for energy, W is for waterproofing, I is for insulation, and P is for protection. So there's the roles of certain parts of fat. Now I want to talk about one kind of lipid. A lipid that you meet at A level. So what we'll do is we'll move this up here. And I want to think about the triglyceride. Now a triglyceride molecule, a type of lipid, triglyceride molecule is made up of, so I'll put here, a triglyceride, which is a type of lipid, is made up of one lot of what's called glycerol and three what we call fatty acids, and that makes a triglyceride molecule. Now let's think about how that actually forms. So I'm not going to draw the structure with all the bonds. Instead, I'm merely just going to do it like this. So CHHOH What I've drawn here is a simplified version, C being carbon, H hydrogen, O oxygen. This here is a simplified version of a glycerol molecule. And then what we have is something that looks a bit like this. Again, H is hydrogen, O oxygen, C carbon. Each fatty acid molecule, see, has this COOH or HOOC, depends how you say it, attached to them. Now, the R group is the hydrocarbon chain that varies between fatty acids. So you can see where I've written here fatty acid, you can get different numbers of the carbons, hydrogens. They all vary, but they all have this COOH end to them. Now, you might be able to spot that and I'll do this in green, if I highlight that part there, you might be able to see that that would give us two H's and an O. We have an OH on the glycerol molecule and an H on the fatty acid. So we can actually form a triglyceride by what's called a condensation reaction. So by removing the H of the fatty acid molecule and the OH from the glycerol, there, and there, we can actually combine these two to form our triglyceride. So triglycerides are formed in what's called a condensation reaction from glycerol and fatty acids. So that's how you form a triglyceride. Now, if one of those fatty acids were removed, if we removed one fatty acid and we replaced that with something called a phosphate, that is what we call, and I put it in brackets here, that's what we call a phospholipid. And that's a different type of lipid. So a triglyceride, and this is a common question student gets asked, the difference between a triglyceride and a phospholipid. A triglyceride contains one glycerol and 3 fatty acids. A phospholipid contains one glycerol, 2 fatty acids, and a phosphate molecule instead. So the phosphate, a phosphate, if we think of it as a little P with an I, so an inorganic phosphate, instead of that fatty acid, that 3rd fatty acid, that would give rise to a phospholipid. Now, a phospholipid is what makes up our cell membrane. So I've done a video already on the structure of a cell membrane, and it's made up, the cell membrane basically is a bilayer of phospholipids, and they look exactly like that. So that's why I included that little picture in the title, lipids, because what I've drawn is a phospholipid. It has a sort of head that's hydrophilic, water-loving, and a little tail, the 2 tails coming down, you can see, which represent our fatty acids, and they're hydrophobic, they're water-hating. So that's just a little bit about the difference between a triglyceride and a phospholipid. Triglyceride has 3 fatty acids, phospholipid has 2 and a phosphate group attached. And we've just said a little bit about the functions of lipids, so energy, waterproofing, insulation, and for protection. Interestingly, if we were to add water, so if we just go back to our triglyceride molecule, if we were to add water to that, that would be a hydrolysis reaction. So if we were to put back the H2s and the O, so the H in the fatty acids and the OH of the glycerol, we would get back our glycerol and fatty acids separately. So in a condensation reaction, to confirm they join together, you get the glycerol joining with the fatty acids. If we were to add water rather than take it away, that would be a hydrolysis reaction, and that would allow us to get our glycerol and fatty acids to separate back into their individual forms. One thing just to add is about the difference between saturated and unsaturated fatty acids. In fact, I'll just do this as a small little bit, just by, so I'll reduce this down here. When you look at fatty acid structures, fatty acids that we call saturated have a C to C bond. So they have carbon atoms joined by single bonds. So if you see single bonds between carbon atoms, we say that is a saturated fatty acid. And a good example of that is stearic acid. All the carbon atoms there are joined by single bonds. If in the molecule you were to see this, the presence of a double bond between two carbons, that would be an unsaturated fatty acid. So unsaturated fatty acids have at least one carbon-to-carbon double bond, at least one carbon-to-carbon double bond. Polyunsaturates have more than one double bond, as in linoleic acid. So that's LINOLEIC, linoleic acid. That's an example of a polyunsaturate. So unsaturated fatty acids have at least one C to C double bond. Saturated fatty acids only have single carbon-to-carbon bonds. So it's worth just mentioning that there. The final question that you might get asked in an exam, certainly you'd be asked functions of lipids, difference between triglyceride and phospholipid, is the test for lipid. Now the test for a lipid is something called the emulsion test. Now, lower down the school at Key Stage 3, when you were testing for fat, you might have got a piece of tracing paper or greaseproof paper. You rub the fat on and you see if it's translucent. Now, that's a little bit basic for what we're talking about now. What you have is the emulsion test. So I'll just talk through this, and that's where this video will finish. So again, we'll reduce this down, we'll put this here. So the test for lipids is known as the emulsion test. We'll talk through the stages of that emulsion test. So, number one, if you wanted to find out if there was any fat in a particular food, you do this emulsion test. So you put the substance that you're testing in a test tube. So we'll colour this blue here. So the substance to be tested, you put into a test tube here, and you'd add to that test tube ethanol. And you shake the test tube with ethanol for about a minute. Then you pour that solution, so once you've shaken that up, you pour that solution into another tube, just for the purposes of distinguishing between the different steps. I'll just do a different colour. So after you've added ethanol, the second stage then is to pour the solution into water. So you put it in ethanol first, really key, then you put it in water. And any lipid would show up as a milky emulsion. So the more lipid there is, the more noticeable the milky colour you would find rather. So when we first put it in ethanol and then you add water to it, what you'll see is a milky emulsion forming if there is lipid present, because we said that lipid doesn't dissolve in water. So on the surface you'd see this milky emulsion forming, and that ultimately is the emulsion test. Take the sample, add ethanol, shake. So it's important, in fact I'll write that over here so you remember. So we shake it up in the ethanol. Once you've shaken in ethanol, you add water to that solution. And then if there's fat present or lipid present, we would get a milky emulsion appearing. And that's what that yellow band just there is. That's a milky emulsion. And that's our emulsion test for lipids. Okay, I hope all that helps.